Hello everyone, and welcome to this audio podcast. This audio podcast is part of an Erythropoiesis edition. This audio podcast is part of a hurricane Plexar. This audio podcast is part of a Labonix Vendip. This audio podcast is part of a Storm Robotics production. This audio podcast will sort of will sort of interconnect the audio podcast series by going into sort of an analysis or a critical review of a critical review in around some of the literature, but also again sort of comparing some of the sort of pharmacotherapies and pharmacodynamics and pharmacokinetics of immunosuppression as part of this audio podcast. So in one investigation, the results at 3 years of recipients who received first cadaveric renal allografts or kidney allografts, and were treated with either tacrolimus or mycophenolate, CSA, L-cyclosporin, and MMF, mycophenolate or tacrolimus, and AZA or azathioprine. 223 recipients were randomized to receive one of three regimens along with corticosteroids. Patients or recipients who experienced delayed graft function or DGF also received antibody induction. Patients or recipients with DGF and ATN or acute tubular necrosis treated with tacrolimus and MMF experienced a 34% increase in allograft survival compared to patients receiving CSA and MMF. P-value was 0.02. The patients or recipients randomized to either treatment containing tacrolimus exhibited superior renal function when compared to CSA. Over the 3 years of follow-up, a new onset insulin dependence occurred in around 6, 3, and 11 recipients in the tacrolimus and MMF, tacrolimus and MMF, CSA and MMF, and tacrolimus and AZA cohorts accordingly. Furthermore, recipients randomized to tacrolimus and MMF received significantly lower doses of MMF as compared to those administered CSA and MMF. It was concluded that all three immunosuppressive regimens provided excellent safety and efficacy. However, the best results overall were achieved with tacrolimus and MMF. This combination may provide particular advantage to renal allografted recipients with DGF with ATN or acute tubular necrosis. In recipients who experienced DGF and ATN, renal function was better at 3 years in 3 years in those recipients receiving tacrolimus in combination with either MMF or AZA as compared with recipients receiving CSA and MMF. When OPALS analyzed the database, the author have found recipients treated with CSA and azathioprine immunosuppression therapy had the best long-term prognoses. followed by recipients on CSA monotherapy. However, retrospective data analysis like this can be criticized because recipient selection bias is a critical problem. Even though many questions concerning immunosuppression with CSA have been addressed, its optimal use has been far from being answered. Only recently, however, again, sort of retrospectively, Kahan and team, Kahan and team, Kahan and team, Kahan and team, and reported that the incidence of chronic rejection was 24% post 5 years for renal allografted recipients with relatively stable pharmacokinetic CSA profile, but 40% in unstable recipients. Hence CSA drug treatment based on 2-hour post-intake blood sample measurements might lessen toxicity and optimizes immunosuppression therapy and potency. There is also evidence that CSA absorption profiling in immediate post-transplantation period reduces the incidence of severity in AR or acute rejection. Another investigation carried out was found previously that pre-transplantation CD4 activity predicts the risk of acute renal transplant rejection, and to analyze the effect of CSA in AZAR, CSA MMF, and FK506 or tacrolimus and AZAR therapy on CD4 helper function, B cell monocyte and cytokine responses. AZAR therapy on CD4 helper function and B cell monocyte and cytokine responses were part of an investigation. 82 renal transplanted recipients were but also where the follow up was 4 months post transplantation. A allogenic co-cultural system was utilized to assess CD4 helper cells to assess the levels of T helper cells and T cell dependent B cell responses. B cell differentiation was assessed in a reverse hemolytic plaque assay. Transplantation immunosuppression therapy in line with transplantation immunosuppression therapy. Pre-transplant CD4 helper defects and low interleukin 10 and enhanced interleukin 4 responses of CD4 cells predicted a low risk of acute rejection. One of the 34 recipients, or around 33, around 30%, 3% of the recipients exhibiting at least one of the 3 low-risk parameters experienced acute rejection, in contrast to 15 to 48 recipients, or 31%, who did not. Only tacrolimus-based immunosuppression therapy resulted in an increased incidence of CD4 helper defects of CD4 helper defects 4 months post-transplant. Post-transplant compared to pre-transplant incidence as well. Post-transplant compared to the pre-transplant incidence was also assessed, and where the p value was mildly significant, or mildly important, was mildly mildly sort of used as part of the assessment, a part of the assessments. At the same time, tacrolimus and AZA treatment was associated with an increase in CD4 cell and B cell interleukin 10 responses, and an occurrence of acute vascular rejection in 3 recipients. 6 patients were switched to tacrolimus and MMF showed no increase in vitro interleukin 10 responses. The investigation concluded that pre-transplant helper defects, low interleukin 10 and enhanced interleukin 4 responses of CD4 cells predict a lower risk of acute rejection. Tacrolimus results in suppression of CD4 helper activity, but CD4 helper activity probably at the same time combined with MMF in immunological high risk paediatrics had been identified as in vitro interleukin 10 responses increased on tacrolimus where tacrolimus and azathioprine treatment were utilized. Another investigation carried to evaluate 2-year results of early CSA withdrawal in sirolimus treated renal allografted recipients. The results had shown at 2 years the incidence of acute rejection with after randomization was not significantly different between recipients who had CSA withdrawn. Who had CSA withdrawn 9.8%, and those who remained on triple therapy 5.1%. Long-term results of CSA elimination with SRL SRL therapy had showed important renal function. Rates of allograft survival 94% versus 91.6%, and prognosis was 95.8%, 95.8% versus 94.9%, were comparable between CSA between CSA, SRL, and SRL, and SRL with a second immunosuppression accordingly. Serum creatinine and systolic ABP was significantly better in SRL recipients. The difference in serum creatinine was consistent and above 30 micromoles for all creatinine quartiles. Long-term results of CSA elimination with SRL showed with SRL therapy showed improved renal function. In another investigation, 120 renal recipients were randomized to receive one of 3 tacrolimus-based regimens tacrolimus-based regimens. Based on low dose, medium dose, or high dose, and where whole blood trough levels were assessed, and also where CSA based therapy oral doses ranging from 3 to 7 milligrams per kilo twice daily. Analysis of the incidences of acute rejection and toxicity within the first days post-transplantation revealed that FK506 or tacrolimus trough concentrations in the range of 5 to 15 nanograms per milliliter were sufficient to achieve optimal efficacy with minimum toxicity. At this optimally defined low dose FK506 regimen, the incidence of biopsy-proven acute rejection in recipients was similar to that of obtained with CSA-based therapy, 18% versus 21%. Moreover, 9% of recipients with low-dose FK506 5 to 15 nanograms per liter trough levels experienced corticosteroid-resistant acute rejection requiring anti-lymphocyte or OKT3 analog treatment compared to none of the CSA treated compared to CSA treated recipients. Comparison of survival and allografts allograft survival of CSA and FK506 in renal transplantation had also been important, where initial investigations with CSA microemulsion were performed to compare pharmacokinetics, clinical efficacy, and safety profiles to that of the original formulation of CSA. In addition to improved and more consistent CSA bioavailability, several investigations demonstrated a significant reduction in incidence of acute rejection in renal and liver transplanted recipients treated with the original formulation. More recent investigation had been initiated to compare clinical efficacy and safety profiles of CSA microemulsion to that of FK506 in both renal and liver transplanted recipients, and in prospective randomized trial of 48 renal transplanted recipients followed up for a minimum of 3 months. Recipient prognoses and allografts prognoses were comparable, with no significant differences in the indices of biopsy proven and steroid resistant acute rejection. And so, while reports on the use of FK506 or tacrolimus in pediatric transplanted recipients are mostly favorable, there have been reports of tacrolimus induced toxicity and the development an increased incidence of post-transplant lymphoproliferative disease or PTLD. Where PTLD had been inferred. In a report by an author Atkinson, retrospectively, FK506 administration resulted in hypertrophic cardiomyopathy in 5 consecutive pediatrics with acute allograft failure, failure, and progressed to CHF in 2 of the cases. Fortunately, the cardiomyopathy regressed in all 5 recipients following conversion to CSA. And because the calcineurin inhibitors CSA and FK506 have a different potential, have a significant potential for nephrotoxicity, novel immunosuppression have been designed to reduce or eliminate toxicity, nephrotoxicity. And sirolimus, again, for example, being a monoclonal, upon the monoclonal antibody, from the monoclonal antibody directed against the alpha chain of interleukin 2R or anti-CD25, similar to monoclonal antibodies like such as daclizumab, alizumab, and basiliximab, and polyclonal antibody thymoglobins. Update on in around this another basis in the low end of sirolimus therapeutic range is or has set to protect against rejection. The high end the high end has been to avoid an unacceptable level of toxicity and other undesirable effects. Several factors must be considered when applying a therapeutic range, a therapeutic reference range. While sirolimus was approved by FDA in combination with CSA and for renal transplanted recipients, there are other protocols. There have been other and therapeutics evaluated, and sirolimus is or had been at the same time where sirolimus or SRL have been evaluated for use in around liver transplantation, possibly other solid organ transplants. Some transplant centers may dose higher than others. The appropriate therapeutic range is dependent on the type of transplant, the combination of immunosuppressants when the patient is in a maintenance phase, as opposed to a loading dose phase. The approach of the transplant center and the analytical modality or method and the analytical method for the current for the current for current and most for the current and most current sort of procedure the in most current procedure in around renal transplantation a combination of CSA given 4 hours before SRL or sirolimus at steady basis steady titer levels, and particularly in a maintenance phase with tests performed at least with for example HPLC uh, a range had been evidenced between 4 to 12 nanograms per milliliter, where the trough level before or should be achieved is applicable, a reference range of 4.0 to 12.0 nanograms per milliliter. Trough level should be achieved between the different doses, and had been retrospectively suggested. All studies carried and stated in part of this critical this sort of critical review have given sort of opinions in around immunosuppression therapies and toxicities. Nephrotoxicity, among many other toxicities that affect pediatrics with renal failure post transplantation, with the use of immunosuppression therapy with the use of immunosuppression therapy is generally unfortunately common. There are several drug regimens today prescribed to pediatric renal sufferers. All have different pharmacokinetic and pharmacodynamic bases, and recipients may be on, for example, MMF or azathioprine as prophylactic suppressant therapies. These drugs are not used in the long term for adequate therapy. Most patients or recipients in the UK are on in the UK but also in other parts of the world, are on CSA or FK 506, and then prednisolone as a corticosteroid. Fewer recipients are on prograph or on FK506, for example, than CSA, are on and at the same time are on a third drug past the two basic. Maintenance therapy will always include CSA, or FK506, and the other drugs are for additional protection but not helpful to the same extent. Thus, SRL sirolimus will be used or has been used like MMF, like a mycophenolate mofetil, and AZA or azathioprine. Many recipients also now on a sort of additional formulation of CSA, and have been better absorbed by um, and nephrotoxicity still being one of the main challenges. However, it's universal with other drugs. However, individual susceptibility plays a role, as do blood levels achieved. The more in the blood, the more the kidney, or the more the renal pathophysiology is effective, or is effective. Trough levels are normally standard procedure in around renal insufficiency. Thus, levels are used to give indication of blood concentrations before to administering doses. These tend to be helpful and give clues to how much drug effect the recipient is getting. But to really get the best results, specialists require the sort of kinetic profile, and look at a drug level every hour or two, doses, and calculate an average blood level. Research has been carried out to find a better way to monitor the drugs. The reason why drug monitoring is important is simply because specialists know that therapy is working, or treatment is working. An organ is being suppressed. An organ is being an organ an organ is sorry it, the tissue or the pathophysiology is being suppressed, to allow allograft functioning. Thus too high levels give nephrotoxicity, and low levels can prompt rejection. And so some of the investigations that have been carried are informative and show a good definition, though to really understand nephrotoxic in children or pediatrics, better research and laboratory procedures are required. It is fair to say that nephrotoxicity occurs later on post-transplantation, thus occurs in a long term, though this does not make it any better for the child or pediatric with renal failure. Because of ill health, the child only wants to live would only want to live, and how he is and based on because of ill health, and where toxicity occurs, where toxicity occurs, no one knows how renal failure sort of sort of develops at for example a part of at sort of DNA at DNA basis, for example, at DNA basis, although prospective research has looked at, for example, single nucleotide polymorphisms or SNPs. But at the same time, in particular, by the biochemical pathways involved and the pharmacokinetics through cells and research around this is still wanting. It has been felt that carrying out with research or investigation in animals more often than not can be irrelevant, because the results will mostly be irrelevant against humans, against humans, because of the immune system is not is the immune system in the main and the immune system in the main being different. Thus, nephrotoxicity investigations need better organization and more relevant cases for its studies. Relying only on serum creatinine increases rather than percent decreases in endogenous creatinine clearance may lead to the relative overdosage of drugs. So, for example, again, sort of aminoglycosides, as well as anti-rejection therapy and nephrotoxicity. The actual measurement or basis of creatinine clearance in hospitalized children may be fraught with problems. So, for example, inaccurate urine collections, incompliant recipients. Incompliant recipients, non-adhering recipients, and need for indwelling and need for example, indwelling catheters, and results necessary to prescribe appropriately or to modify the dose of drugs, or 24 to 36 hours, and completion of urine collections. Thus, other more practical modalities should be utilized. That draws this audio podcast to a close. This has been an audio podcast as part of the Erythropoiesis edition. An audio podcast as part of a Hurricane Plexar. An audio podcast as part of a Labonix Vendip. An audio podcast as part of this series. Where this audio podcast was a sort of a critical review, looking at sort of different immunosuppression. Thank you.